Welcome, dear friend, to Dreamtime Science. Imagine yourself amidst shelves of ancient books, their spines worn soft under the gentle glow of a bedside lamp. The night outside stretches quietly, stars whispering old secrets through the window. Here, in this calm nook, we invite you to drift gently into stories wrapped in tranquility and wonder. If this moment feels right, a quiet tap of the like button or a soft word shared in the comments about where you listen from would mean the world. Let yourself settle now, adjust your pillow just so, breathe in slow, even rhythms. Together we will wander softly through the twilight realms of knowledge, letting curiosity soothe the mind while sleep tiptoes closer. Whenever you are ready, let your journey begin as we turn the page together. Tonight we'll glide very slowly, very gently through the quiet miracle of how a single tiny cell begins its wondrous journey to become a delicate thinking brain. Like a soft whisper unfolding in the hush of twilight, we'll hold a safe, pillow-soft distance, inviting gentle curiosity to drift alongside us. Let your mind wander softly as we begin this peaceful exploration. If this peaceful blend of science and serenity helps ease your evening, consider joining our community of gentle learners. Share your favorite moment of scientific awe in the comments. Before we begin, create your sanctuary. Nestle into your chosen spot, let your muscles soften, and allow your breath to find its natural rhythm. In the hush of late evening, when the world settles into velvet quiet, the mind often drifts to its own mysteries. We are creatures of thought and sensation, wreathed in questions as ancient as dreams. How did consciousness arise? What secret journey transforms a single cell into the luminous storm of awareness that inhabits each of us? The tale begins far before memory, in darkness and warmth, where potential gathers and shapes itself into the beginnings of a mind. Let us linger a moment at the crossroads of science and story, where philosophy and fiction have long wrestled with the riddle of the self. Films like The Matrix and Ghost in the Shell conjure visions of consciousness severed from flesh, minds floating in virtual spaces, identity untethered from biology. They ask, what are you, if not your brain? They tempt us with digital dreams, the possibility of uploading a soul or awakening to discover that all you know is but a simulation. These stories gnaw at the edges of our understanding, but before we can contemplate the mind untethered, we must first walk the path of its becoming, the astonishing odyssey that begins with a single fertilized cell. Consider the zygote, a speck of life, more promise than form, formed when sperm and egg meet in the shrouded corridors of the womb. It is, at first, a sphere of possibility, a blueprint inscribed not with ink, but with the intricate language of DNA. This cell seems humble, almost unremarkable, a dot among billions. And yet, within its fragile membrane, the future hums. It carries within it the ancient instructions passed down by unbroken chains of ancestors, the distilled wisdom of survival, adaptation, and, ultimately, awareness. The zygote divides once, then twice, then many times more in a silent, rhythmic dance. With each division, the cells begin to specialize. They whisper to one another with molecular signals, orchestrating the first steps of a grand ballet. Some will become skin, fragile and translucent. Others will form the heart, which will beat long before the world is seen. And from a slender strip of cells along the embryo's back, called the neural plate, will arise the beginnings of the brain and spinal cord. This is the genesis of consciousness unfolding in the secret theatre of the womb. The neural plate folds, curling and arching, until its edges meet and fuse. Thus is formed the neural tube, a delicate channel from which the entire central nervous system will emerge. At one end, the tube swells, ballooning into the three primary vesicles that will become the forebrain, midbrain, and hindbrain. Here, in these bulges and folds, the architecture of the mind is laid out in miniature, an echo of evolutionary history, replayed in fast forward. The raw material of the brain is not stone or metal, but living tissue, cells known as neurons, which will soon reach out with filigreed arms called axons and dendrites. These cells are both architects and masons, laying down the scaffolding for thought, sensation, and memory. By the time the brain is recognizable, its neurons are proliferating at a dizzying rate. In the fetal brain, hundreds of thousands are born each minute, a silent surge of creation. The unfurling of the brain is a story written in layers. The earliest structures form deep within, responsible for the most basic functions: heartbeat, breathing, the regulation of temperature. These ancient systems are inherited from our animal ancestors, a testament to life's continuity. Upon this foundation rises the limbic system, the seat of emotion and instinct, and finally the neocortex, a thin, rippled sheet that will cradle the higher faculties of reasoning, language, and imagination, the very crown of the mind. Within the womb, the developing brain is swaddled in darkness, yet it is not idle. Even before birth, the fetus moves, responds to touch, and in the later months is startled by sudden sounds. There is evidence that the unborn child hears the muffled music of its mother's voice, the cadence of language, the steady drum of the maternal heartbeat. Patterns are laid down, rhythms are learned, and the architecture of future perception is shaped by these first shadowy experiences. If we pause here and look more closely, we see how this journey is not only a marvel of biology, but also a crucible of complexity. Each neuron must migrate to its appointed place, guided by chemical signals like travelers following invisible beacons. Once settled, they extend their branches, seeking partners in an intricate dance of connection. Synapses, the tiny gaps where signals leap from cell to cell, are formed in a profusion that staggers the imagination. At birth, an infant's brain contains more synapses than there are stars in the Milky Way, a vast and tangled jungle of possibility. It is here that myths arise, born of awe and misunderstanding. You may have heard the oft-repeated claim that we use only 10% of our brains, a notion as persistent as it is misleading. The truth is subtler, more wondrous. Even in the quietest moments, nearly every region of the brain is active, humming with the silent chatter of billions of cells. No neuron is wasted. The brain is not a dim, half-lit mansion, but a city ablaze with energy. Its districts alight with sensation, memory and thought. Where, then, did this myth originate? Perhaps it sprang from the observation that at any given instant not every neuron is firing at once. Or it may have emerged from early neuroscientific explorations, when much of the brain's function was still shrouded in mystery and uncharted territories were mistaken for emptiness. Yet with the advent of modern imaging, functional MRI and PET scans, we see the truth made visible. Even the simplest act, such as reading these words, stirs a symphony of activity across the brain's landscape. In the stories spun by science fiction, the brain is often depicted as a programmable machine, a substrate for consciousness that might be swapped, copied, or erased. The Matrix envisions minds suspended in digital fantasy, their bodies mere batteries for an artificial dream. Ghost in the Shell imagines cyborgs whose consciousness can be uploaded, manipulated, or even stolen. These tales are alluring, but they gloss over the profound, embodied complexity of our true origins. The brain is not an island adrift in the skull, but a living organ shaped by the body it serves, the world it inhabits, and the history it carries. In the warm dark of the womb, the brain's development is inseparable from the pulse of the mother's blood, the ebb and flow of nutrients, the gentle rocking of the amniotic sea. The fetus is not a passive passenger, but an active participant, its movements shaping the growth of muscles and bones, its first neural firings guiding the sculpting of circuits that will one day give rise to consciousness. Experience, even before birth, is woven into the fabric of the growing brain. As the pregnancy progresses, the brain's surface begins to fold and crease, forming the characteristic gyri and sulci that allow its vast surface area to fit within the confines of the skull. These folds are not mere decoration, but the signature of complexity. The more convoluted the cortex, the greater the potential for intricate networks of association and memory. In these furrows and ridges, the seeds of individuality are sown. The tale of the brain's genesis is also a story of vulnerability. The same chemical signals that guide neurons to their proper places can be disrupted by chance or by harm, by genetic mutation, by toxins, by malnutrition. The developing brain is exquisitely sensitive, a masterpiece in progress that can be shaped or scarred by the environment. Yet it is also resilient, capable of rewiring and adapting, finding new paths when old ones are blocked. This plasticity, this capacity for change, is the source of both our strength and our fragility. In the final weeks before birth, the fetal brain undergoes a surge of growth. Myelin, a fatty substance, begins to sheath the axons, insulating them and allowing signals to travel with lightning speed. The first patterns of sleep and wakefulness emerge, a rehearsal for the cycles that will govern life outside the womb. The senses, once dormant, flicker to life. The fetus turns toward light, startles at loud noises, tastes and swallows the amniotic fluid. The world is still distant, but the brain is reaching out, preparing for the flood of sensation that will come with birth. Somewhere in this tumult of growth and connection, consciousness stirs. It is not a single moment, but a dawning, a gradual emergence, like the slow brightening of a sky before sunrise. The brain, once a cluster of undifferentiated cells, now hums with activity, circuits firing in patterns that will one day give rise to perception, memory, and the ineffable sense of self. This is the genesis of awareness, the first faint glimmer of the mind's light. Yet the journey is far from over. Birth is not an end, but a beginning. The newborn brain is unfinished, its circuits awaiting the sculpting touch of experience. The world will shape it, carving new pathways, pruning unused connections, strengthening those that are called upon again and again. The story of consciousness is not written in the womb alone, but in every moment that follows, each sensation, each encounter, each memory. We might return for a moment to those tantalizing visions of science fiction. The notion of the brain as a computer, consciousness as code, is a powerful metaphor. It speaks to our yearning for mastery, for the ability to transcend flesh and bone, to become something more than the sum of our parts. But the truth is more wondrous and more humble. Consciousness arises not from silicon or circuitry, but from the ceaseless interplay of biology and experience, from the dance of cells and the whisper of memory, from the pulse of blood and the breath of air. Within each of us is the echo of that first dividing cell, the memory of the neural tubes folding, the legacy of 1,000,000 migrations and connections. We carry the history of life itself, written not in books but in the living architecture of our brains. And as we ponder these origins, we are reminded that the mind is not a static thing, but a process, an unfolding, a becoming, a story still being written. In the quiet that follows, as the mind drifts toward rest, there lingers the wonder of it all, that from a single cell, guided by the silent choreography of molecules, can arise the miracle of consciousness. The brain, that most mysterious of organs, is at once a product of biology and a vessel for the ineffable. Its genesis is a story of possibility, of fragility and strength, of the endless dance between matter and meaning. And so, as night deepens and the world grows still, the mind turns gently to the next chapter in its story, a story that will lead us from the quiet beginnings in the womb to the first moments of awareness, the first encounters with the world, and the lifelong journey of the thinking self. The genesis of consciousness is but the opening movement, the soft prelude to a symphony not yet played to its end. Within the sheltered vault of the skull, in that dim-lit labyrinth of grey and white, the story of the mind unfolds in a choreography that is as mysterious as any dream. If you could shrink yourself to the size of a dust mote and journey through the meandering corridors of the brain, you would discover a landscape more complex than any city, more ancient than any myth. It is here, in this living maze, that the raw stuff of consciousness is spun, a process as profound as it is enigmatic. Let us begin at the threshold of becoming, where the journey of a single cell sets the course for destiny. Deep within the developing embryo, a host of identical cells hum with potential. Yet at some pivotal moment, a chemical whisper, a jostle of neighboring cells, a gradient of signaling molecules, some of these cells make a fateful choice. They embark upon a path that will see them transformed into neurons, the signal bearers of the nervous system, or glial cells, the silent guardians and architects of the brain. This decision is orchestrated by an intricate ballet of genes and proteins, a code written in the language of DNA, but interpreted through the shifting symphony of the cellular environment. How does a cell know what to become? The answer, while grounded in molecular biology, has the air of a riddle. Within the cell's nucleus, DNA holds vast libraries of instructions, but not every chapter is read at once. Chemical tags, methyl groups, acetyl groups are fastened to the genetic script, opening some passages for transcription while sealing others shut. Signals from neighboring cells, perhaps diffusing proteins or subtle electrical cues, prompt a cascade of changes. Certain genes awaken, others are silenced. This is the art of differentiation, where a cell's fate is sealed not by a single command, but by a dialogue, one that weaves together the cell's lineage, its environment, and the stochastic role of molecular dice. For neurons, the path is extraordinary. Once committed, a young neuron begins to grow, extending a slender process, the axon, across vast distances, searching for its future partners. Along the way, it sprouts fine, branching dendrites, receptive to the molecular echoes of other cells. Meanwhile, glial cells, often relegated to the background in popular imagination, play roles no less vital. Astrocytes, with their star-like arms, nourish neurons and modulate synaptic communication. Oligodendrocytes wrap axons in fatty myelin, speeding the transmission of electrical impulses. Microglia patrol the tissue, guardians against infection and decay. In this manner, the brain's architecture is built, a cathedral raised from the collaboration of many kinds of cells, each with its own whispering voice in the symphony of mind. Yet structure alone is not the mind. The true wonder emerges in the connections. Each neuron may reach out to thousands, even tens of thousands of others, forming a network so dense that if you unraveled the wiring in a single human brain, the filaments would stretch for hundreds of thousands of kilometers. At the junctions, synapses, chemical messengers leap across infinitesimal gaps, translating the electrical language of one neuron into the chemical dialect of another, only to be rendered electrical once again. This is not a static arrangement. The connections themselves are forged, strengthened, or pruned over time, guided by experience, by use, by the mysterious algorithms of learning and forgetting. Consider for a moment the paradox this presents. The signals that course through the brain are physical, ions flowing across membranes, neurotransmitters drifting between cells, proteins assembling and disassembling in response. And yet, from this physicality arises something ungraspable, the sensation of a memory, the pulse of an emotion, the flicker of a dream. If you have ever watched Christopher Nolan's Inception, you know the allure and peril of descending into layered realities, each one nested within another. The brain, too, is a hall of mirrors, where perception and recollection, emotion and reason all interleave in ways that defy simple mapping. Within the hippocampus, a seahorse-shaped structure deep within the temporal lobe, new memories are etched into the neural fabric. Here, a unique population of neurons is born even in adulthood, a rare example of ongoing neurogenesis. The hippocampus acts as a gateway, encoding experiences and relaying them to be stored elsewhere, perhaps in the vast association cortices of the neocortex. But the memory is not a static engraving. It is more like a melody played on a piano, reconstructed anew each time it is recalled. Synaptic weights shift, new branches form or retract. The memory is revised and reshaped by the very act of remembering. Like the shifting architecture of dreams in Inception, the landscape of memory is always in flux, never quite the same from one visit to the next. Even more elusive is the question of consciousness. How does the firing of billions of neurons, the ceaseless interplay of molecules and ions, give rise to the inner world, the feeling of I? Science has mapped the highways and byways of sensation, perception, and action, but the subjective experience itself, the qualia, as philosophers call it, remains outside the grasp of experiment. Some researchers liken consciousness to an emergent property, like the wetness of water arising from countless H2O molecules, or the patterns in a murmuration of starlings. Others posit that certain kinds of information integration or particular patterns of network activity are the seat of awareness. Yet as in the film Lucy, where the protagonist's brain capacity is gradually unlocked, we are left to wonder, is consciousness a matter of degree or of kind? Are we merely glimpsing the surface of a vast ocean, unaware of the depths below? The structure of the brain itself reveals hints, if not answers. Folded and convoluted, the cerebral cortex is a sheet of tissue just a few millimeters thick, yet it contains billions of neurons arranged in layers and columns. In some regions, the columns process the raw data of vision, turning points of light into edges, shapes, and colors. In others, they conjure language, or orchestrate movement, or spin the stories that make up the self. Beneath the cortex, older structures, thalamus, basal ganglia, amygdala, mediate emotion, attention, and the rhythms of sleep and waking. The brainstem, ancient and robust, keeps the heart beating and the lungs inflating. Each region, specialized and yet interconnected, contributes its own melody to the orchestral whole. The connections between regions are as important as the regions themselves. White matter tracts, insulated highways of axons, carry signals across the brain with astonishing speed. The corpus callosum, a great bridge of fibers, links the two hemispheres, allowing the left and right sides to share information. Disrupt this bridge, and the mind itself can seem divided. Split-brain patients offer a glimpse into how unity of consciousness is constructed from the dialogue of parts. Within this labyrinth, patterns emerge and dissolve. The default mode network, a constellation of regions that hums with activity when the mind is at rest, only to quiet when focused attention is required. The salience network, ever vigilant for changes in the environment. The executive network, marshalling resources for planning and decision-making. These are not static structures, but dynamic alliances, coalitions of neurons rising and falling in synchrony, shifting with mood, task, and fatigue. In moments of inspiration, networks that are usually segregated may briefly unite, sparking the creative leaps that define art and science. In the throes of sleep, these networks decouple and recombine, giving rise to the surreal narratives of dreams. The boundaries between waking and dreaming, between perception and imagination, are porous, ever-shifting. The brain is not a static engine, but a living, breathing landscape, forever remapping itself in response to the outer and inner worlds. At the smallest scale, down among the molecules and ions, another layer of complexity is at play. Each synapse, that minuscule gap between neurons, is a theater of possibility. Neurotransmitters, glutamate, GABA, dopamine, serotonin, are released in precise quantities, binding to receptors, opening channels, allowing a rush of calcium or sodium or chloride. The balance of excitation and inhibition, the timing of spikes and pauses, encodes information in patterns that unfold over milliseconds, yet the same synapse can be strengthened or weakened over hours, days, or years, depending on how it is used, a phenomenon known as synaptic plasticity. This is the molecular substrate of learning, the ever-shifting foundation upon which memory and skill are built. Sometimes the system falters. A mutation in a single gene, a misfolded protein, a rogue immune cell, these can unravel the delicate balance, giving rise to epilepsy, schizophrenia, or neurodegeneration. The brain's complexity is both its strength and its vulnerability. Like the dream worlds of Inception, exquisitely detailed but fragile, a small disturbance can ripple outward, distorting perception, emotion, or will. And yet, even in the face of injury or loss, the brain is resilient. After a stroke, surviving neurons may sprout new connections, rerouting functions to undamaged areas. In blindness, the visual cortex can be recruited for other senses, sharpening touch or hearing. The labyrinth is not fixed, but alive, capable of self-repair and reinvention. All the while, the cells of the brain live and die, their fates entwined with the choices made long ago in embryonic development. The question of how a cell knows to become a neuron or a glial cell is thus not merely a technical one, but a meditation on potentiality and destiny. The same can be said for the connections these cells form. It is not a simple matter of wiring up a circuit, but an ongoing negotiation, guided by genes, sculpted by experience, and always subject to chance. If you listen closely, you can almost hear the whisperings of this negotiation, the faint crackle of synapses firing, the shifting tides of neurotransmitters, the slow remodeling of connections as a memory forms or a habit is broken. It is as if the brain is always dreaming, even in waking, reshaping itself with every thought, every sensation, every encounter with the world. Pop culture, in its own way, gestures toward these mysteries. In Lucy, the premise that we use only a fraction of our brain's capacity is more myth than fact, but it hints at the vastness of the unknown, the sense that there are powers within us yet untapped, realms of cognition and perception that we have only begun to explore. Inception's dream architects, building worlds within worlds, echo the recursive nature of self-awareness, the way the brain models not just the world but itself within that world. The deeper we delve, the more we sense the limits of our understanding, the shadowy corridors that remain unexplored. We are left then wandering these halls, marveling at the intricacy of the brain's design, the elegance of its function, and the enduring enigma of its self-awareness. Each discovery opens new doors, reveals new passages, and with each answer a dozen new questions arise. What is the true nature of memory? How does the mind emerge from matter? Where does the labyrinth end? And is there at its center a final secret waiting to be revealed? For now we drift onward, deeper into the maze, guided by the faint glow of insight, the pulse of curiosity, the quiet certainty that wonders yet unknown lie just ahead, waiting in the next fold, the next synapse, the next dream. In the deep hush of night, let us drift into the labyrinth of inquiry that is the human brain, not just the brain itself, but the winding, centuries-long journey undertaken by those who have sought to decode its mysteries. The mind, with all its flickering thoughts and elusive dreams, sits nestled within the soft folds of the brain. To untangle this most intricate of enigmas, we must first turn our gaze away from the present's blinking machines and peer backward into the candle-lit labs and curious cabinets of the past. Here, the science of the brain began not with certainty, but with wonder and often error. Long before the word neuroscience was etched into textbooks, the earliest thinkers puzzled over the seat of consciousness. Was it the heart, thumping with emotion, or the brain, silent and secretive? The ancient Egyptians, meticulous in their preparations for the afterlife, removed the heart with reverence from the bodies of their dead, but scooped out the brain with little ceremony, seeing it as little more than cranial stuffing. It would be centuries before the brain was recognized as the throne of thought. With the passage of time, curiosity sharpened into investigation. By the late 18th and early 19th centuries, a peculiar science called phrenology took root. Its practitioners, often equipped with polished skulls and measuring calipers, believed that the shape of your head could reveal the contents of your character. They mapped the scalp into regions, organs, each said to govern a particular trait: benevolence, combativeness, hope, or secretiveness. A bump behind the ear, according to these maps, indicated a penchant for poetry. A dip along the brow might betray a lack of musical talent. To our modern ears, this sounds fanciful, even ridiculous. Yet in its day, phrenology was a scaffold upon which many earnest thinkers hung their hopes of understanding the mind. Phrenology's greatest contribution, ironically, was not in what it got right, but in what it inspired. The notion that the brain might have specialized regions, however naively mapped, planted a seed. It suggested that the mind was not a single, undifferentiated essence, but a mosaic of faculties, each potentially tied to a distinct patch of neural landscape. The idea was flawed, but it pointed the way. As the 19th century unfurled, new tools arrived. The scalpel and the microscope became allies in the hands of anatomists who sliced through the grey folds of the brain, sketching its valleys and ridges in precise detail. Legends arose around figures like Paul Broca, who in a Parisian hospital encountered a patient known as Tan, named for the only syllable he could utter. Upon Tan's death, Broca examined his brain and found a lesion in the left frontal lobe. This patch, Broca's area, seemed tied to the power of speech. Soon after, Carl Wernicke identified another region, this one linked with understanding language. The brain, it became clear, was a territory of hidden provinces, each with its own dialect and duty. A parallel drama unfolded in the world of electricity. In the late 18th century, an Italian scientist named Luigi Galvani discovered that a frog's leg would twitch when touched by a spark. Was this the vital spark that powered thought? By the 19th century, the idea that the brain might operate through tiny electrical signals took hold. In dim laboratories, researchers placed electrodes on exposed brains, thrilling to the sight of a limb jerking or an animal's eye blinking in response. The true magic of the brain, though, was still just out of reach. How could you study the living mind without destroying it? How could you peer into thought itself? For much of history the answer was, with great difficulty and often by accident. One of the most famous accidents in neuroscience unfolded on a dusty railroad in Vermont. Phineas Gage, a young foreman, was tamping down gunpowder with an iron rod when the charge exploded, sending the rod hurtling through his skull. Miraculously, Gage survived, but those who knew him claimed he was never the same. Once diligent and polite, he became erratic and impulsive. The rod had passed through his frontal lobes, and in the strange aftermath of his injury, the world glimpsed the profound ties between brain structures and personality. The brain was not just a bundle of nerves, it was the crucible of self. As the 20th century dawned and the electric hum of modernity grew, so too did new ways of peering into the brain's hidden chambers. The microscope revealed not just gross structures, but the tiny, branching forms of neurons. cells that communicated across minute gaps called synapses. Santiago Ramón y Cajal, the Spanish father of modern neuroscience, captured these cells in delicate ink drawings, their filigreed webs hinting at the immense complexity within each brain. Each neuron, he realized, was a universe unto itself, sending tendrils into the darkness to touch its neighbors. Communication was not continuous, but a flicker from cell to cell, a process as fleeting as fireflies in a summer night. Yet still, to understand the brain in action, to witness thoughts as they flickered into being, scientists needed new inventions. The quest led to machines that could listen for the faintest whispers of electricity or capture the subtle heat of metabolism. The first of these, the electroencephalogram, better known as the EEG, was devised in the 1920s by Hans Berger, a German psychiatrist. Berger recorded the faint, rhythmic waves that rose and fell across the scalp like the tide brushing a distant shore. Each brainwave, he discovered, reflected some aspect of consciousness, alpha rhythms during quiet wakefulness, delta waves in the depths of sleep. The EEG could not show you where a thought formed, but it could record the music of the mind as it played. In time, the technologies grew ever more refined. The 1970s and 80s brought the arrival of computed tomography, CT, and magnetic resonance imaging, MRI. CT scans, using X-rays to build up a three-dimensional portrait, could reveal tumors and strokes in exquisite detail. MRI, with its humming magnets and radio pulses, conjured images of the living brain without a single incision. Here at last was a way to see the brain whole and uninjured, to observe its architecture in the living. But perhaps most wondrous of all was the advent of functional imaging. With positron emission tomography, PET, and functional MRI, fMRI, scientists could watch the brain at work. As a volunteer solved puzzles or gazed at a photograph, regions of the brain would momentarily glow with increased blood flow. Traces of thought written in the language of oxygen and sugar. The fMRI in particular allowed us to see the mind in motion, to map the shifting patterns of activity that accompany memory, emotion, and even dreams. If you have seen the animated film Inside Out, you have glimpsed a playful vision of this complexity. Emotions personified, joy, sadness, anger, fear, jockey for the controls within the headquarters of a young girl's mind. Each memory, each experience is colored by these shifting internal states. The film, though fanciful, hints at a truth uncovered by modern neuroscience. Our brains are not static, but ever-changing mosaics, with networks that light up and subside depending on context, mood, and memory. There is no simple center for happiness or fear. Instead, these emotions emerge from the interplay of many regions, each contributing its melody to the symphony of the self. Yet for all the dazzling images produced by these machines, for all the glowing scans and colored graphs, the brain retains its air of mystery. A photograph can show us where blood flows, but not why a thought arises. A wave on an EEG can reveal the signature of a seizure, but not the substance of a dream. The deeper we peer, the more we realize that the brain is not merely a machine, but a living city, each neuron a citizen with its own role, its own story. To appreciate the full wonder of brain science, it is worth imagining, as in the classic film Fantastic Voyage, what it might be like to shrink down and travel through the folds and caverns of the brain itself. Picture yourself miniaturized to the size of a blood cell, swept along the endless rivers of the circulatory system, through the whispering forests of dendrites and axons. Here, at the synaptic cleft, chemicals leap across tiny voids, carrying messages of pain, pleasure, memory, and motion. A single neuron, with its 1,000 branches, is a world unto itself, but it is only in their ceaseless conversation with millions of others that the mind emerges. As our tools have grown more subtle, so too have our experiments. In the late 20th century, scientists turned to clever designs to peer into the workings of the brain. They studied patients with split brains, individuals whose corpus callosum, the broad bridge connecting the left and right hemispheres, had been severed to treat severe epilepsy. These patients behaved as though they housed two minds in one body. One hand might button a shirt while the other unbuttons it. When an object was placed in their left hand, they could not name it, for the language centers in the left hemisphere had no information about what the right hand held. Such experiments revealed not only the specialized talents of each hemisphere, but the delicate balance that allows a single, unified consciousness to arise. Other scientists have delved into the realm of brain plasticity, the astonishing ability of the brain to rewire itself in response to injury or experience. In laboratories filled with the scent of coffee and the hum of computers, researchers have mapped how musicians develop larger auditory areas, or how the blind learn to see with their fingertips. The brain, it seems, is less like a fixed machine and more like a living ecosystem capable of adaptation and growth. Today, as we gaze at glowing screens and scroll through the latest headlines of mind-reading devices and brain-computer interfaces, it is easy to forget how far we have come from the days of phrenology and skull bumps. The tools have changed, but the questions remain. How does a single cell, dividing and specializing, give rise to thought and feeling? How do billions of neurons, each firing in patterns too complex to fathom, yield the singular experience of being alive? Each answer, hard-won and incomplete, pushes us further into the unknown. As we probe deeper with electrodes, lasers, and quantum sensors, we find not closure but new mysteries. The brain resists simple explanations. It is as much a product of its history as its biology, shaped by evolution, experience, and the ceaseless dance of molecules in the dark. In the quiet of the night, as you listen to the gentle click and whir of your own thoughts, remember that you are both the subject and the observer of this great adventure. The story of decoding the brain is still unfolding, its chapters written in the language of curiosity and wonder. The more we learn, the more we see that the brain is not just an object to be studied, but a living testament to the power of inquiry itself. And as we press onward through the shifting shadows of knowledge, another question beckons. What, after all, does it mean to be conscious? What is the nature of the self that watches, wonders, and dreams? The journey is far from over, and the next step leads us even deeper into the heart of the mystery, where the mind meets the world, and the question of consciousness awaits, softly shimmering in the darkness. It is night. The hush of the world outside settles over you like a gentle blanket, muffling the day's ambitions and anxieties. In this quiet, you may sense the subtle stirrings of your own mind, a flicker of thought, a half-remembered dream, the echo of a memory far older than your conscious self. In this hush, let us drift together, not away from the world, but deeper into it, to the luminous sea of consciousness itself. The brain, as we have witnessed, is a marvel of intricate growth and ceaseless change. It is built from ancient blueprints, yet shaped anew by every fleeting moment of sensation, every glance, every embrace, every sorrow and delight. Billions of neurons, woven together by trillions of connections, create the shimmering tapestry of our living selves. Yet for all our anatomical diagrams and genetic maps, the true miracle of mind eludes the grasp of dissection. It is as if we can study every thread in a tapestry, but still find ourselves astonished when the image emerges alive with meaning. What is it then to be conscious? What is it to know that you exist, to feel the world not just as a sequence of signals, but as a private, continuous unfolding of experience? For the brain is not merely a machine for collecting information, nor even for steering limbs and uttering words. It is the wellspring of awareness, the mysterious process by which electrical storms and chemical rivers in our heads become the vivid theatre of sensation, thought, and self. Philosophers through the centuries have circled this question as moths around a flame. Some have spoken of mind as a ghost in the machine, a substance quite apart from flesh. Others have insisted that consciousness is illusion, a trick of pattern and computation that only pretends to be real. And yet, as you listen, you may notice the warmth of your own breath, the pressure of the pillow, the slow drift of reverie, proof, if any were needed, that consciousness is not so easily dismissed. Carl Sagan, reflecting on the images of Earth captured from the far reaches of space, spoke of our planet as a pale blue dot suspended in a sunbeam. In that cosmic perspective, all the drama of human history, every conqueror and prophet, every joy and heartbreak, shrinks to a mote of dust in the vastness. But in another sense, the very fact that we can contemplate our smallness is itself a kind of miracle. The universe has become aware of itself through the fragile lens of our minds. This is the paradox. The brain arose from the blind workings of evolution, shaped by the struggle to survive in a world of predators and famine and change. Yet out of that scaffolding of necessity there emerged the capacity not only to sense and act, but to wonder, to reflect, to imagine. The same neural circuits that once helped our ancestors track the movement of prey now allow us to ponder the birth of stars, the death of galaxies, the meaning of love and loss. In the privacy of your thoughts, you may sometimes catch the fleeting sense that your mind is both infinitely vast and impossibly small. Vast because it can conjure worlds from nothing, can leap from the taste of a childhood meal to the structure of a distant nebula in a heartbeat. Small because it is hemmed in by the narrow boundaries of sensation and memory, by the limits of language and the slow erosion of forgetting. To be conscious is to live forever at the edge of the unknown, reaching out in hope and curiosity, but always, in some sense, alone. Yet perhaps we are not quite so solitary as we seem. For all our private reveries, consciousness is also a bridge, a way of sharing our inner world with others. Through language, through art, through music and gesture and touch, we send signals across the chasm that separates one mind from another. The brain's development is not a solitary journey, but a dance performed in the presence of others. From our earliest moments, we are shaped by the gaze of caretakers, the sound of voices, the intricate choreography of smiles and frowns and laughter. Our neurons fire in sympathy with the emotions of those around us. Our stories are woven together until it is difficult to tell where one mind ends and another begins. This, perhaps, is the secret at the heart of consciousness. It is both singular and collective, both fiercely individual and deeply communal. Your thoughts are yours alone, yet they are also shaped by the language you speak, the music you love, the ideas you inherit, the touch of those who have cared for you. In every mind a multitude, in every life the echoes of countless others. And what of the mystery itself? The question of why the physical stuff of the brain should give rise to the shimmering richness of experience. For all the advances of neuroscience, the so-called hard problem of consciousness remains unresolved. We can trace the pathways of perception, map the circuits of memory, even predict the flicker of recognition in the cortex. But the leap from matter to mind, from the firing of neurons to the red of an apple, the ache of longing, the hush of awe, this remains, for now, beyond the reach of equations. Some have likened consciousness to a great mirror, in which the universe reflects upon itself. Through the lens of your own awareness, the world becomes present, alive, meaningful. Each moment of perception is a ripple in the sea of being, a fleeting intersection of the outer and the inner. When you gaze at the star-flecked sky, when you listen to music or lose yourself in a story, you are not merely a spectator, you are a participant in a grand unfolding, a node in the web of existence. It is tempting, in moments of solitude or sorrow, to see consciousness as a burden, a fragile flame flickering in the darkness, easily extinguished. Yet there is another way to see it, as a gift, a rare and precious thing shaped by eons of change, sustained by the delicate machinery of cells and molecules, but reaching far beyond mere survival. The brain's development is a kind of alchemy, transforming the brute facts of biology into the gold of meaning. Think of the newborn, eyes wide, drinking in the world for the very first time. In those early days, the brain is a landscape of raw possibility, a chaos of unformed potential. Each sensation, each caress, each lullaby is a thread woven into the tapestry of the self. And as the child grows, as language blooms and memory deepens, a person emerges, a unique perspective, never before seen, never to be repeated. The journey from embryo to adult is not only a biological unfolding, but a voyage into the labyrinth of consciousness itself. And yet, for all our sophistication, for all the marvels of learning and intellect, there is a humility that comes from contemplating our place in the cosmos. Sagan's pale blue dot is a reminder that we are, in the final reckoning, tiny and fragile, adrift in a vastness that is indifferent to our hopes and fears. But it is also a reminder that in the brief moment of our lives, we possess something astonishing, the ability to know, to care, to wonder. What might it mean, then, to be conscious in such a world? Perhaps it is to be a witness, a storyteller, a bearer of memory. The brain, with all its complexity, is not merely a calculating engine, but a vessel for meaning. It allows us to remember those we have loved and lost, to imagine futures yet unborn, to create beauty where none existed before. It allows us to ask questions for which there may be no answers, to explore mysteries that may never be solved. The sea of consciousness is not always calm. There are nights of confusion, days of sorrow, moments when the self seems to dissolve into chaos. Illness and injury can cast shadows across the mind, distorting perception, fragmenting memory. And yet even in these storms there is a resilience, a capacity to heal and adapt, to find new pathways when old ones are lost. The brain, like the ocean, is both fragile and enduring, shaped by forces seen and unseen. In the silence of the night, you may feel yourself drifting on this sea, carried by currents you did not choose, yet able to steer in small ways by the rudder of attention and intention. The act of noticing, of paying attention, is itself a kind of creation. When you listen to the wind, when you taste a ripe fruit, when you remember the laughter of a friend, you are not merely receiving data, you are participating in the unfolding of your own mind, shaping the contours of your inner landscape. There is a kind of peace in recognizing the limits of our understanding. The mystery of consciousness is not perhaps a puzzle to be solved once and for all, but an invitation to explore, to wonder, to cherish the miracle of being alive. The brain gives rise to mind, but mind in turn gives meaning to the universe. Through your eyes the world becomes bright. Through your heart it becomes dear. And so, as you lie in stillness, the rhythms of your breath mingling with the slow tide of thought, you may sense yourself as both very small and very great. Small in the face of the universe's immensity, great in the depth and subtlety of your own awareness. You are the product of ancient stars, the inheritor of countless lives, the bearer of a consciousness that is at once unique and universal. The journey of brain development from a cluster of dividing cells to to the full flowering of adult mind is a story written in molecules but read in experiences. Each person, each consciousness is an unrepeatable moment in the history of the cosmos, a brief flare of selfhood, a spark of awareness, a voice in the chorus of existence. Somewhere far above, the planets wheel in silence. The stars burn cold and distant, indifferent to our joys and sorrows. Yet here, in the warmth of life, in the fragile circuitry of your own mind, the universe awakens to itself. The atoms that once drifted in darkness now dream, now remember, now reach out in wonder. There are questions that linger, as persistent as the tides, questions about the nature of self, about the boundaries of consciousness, about the possibility of minds other than our own. Could there be other forms of awareness hidden in the depths of the sea or the silence of alien worlds? Is consciousness a rare accident or the inevitable flowering of complexity? Such questions may never be fully answered, but they are, in themselves, a testament to the power and mystery of mind. To ask is to reach beyond the given, to imagine what might be, to honor the unknown. In the end, perhaps the greatest gift of consciousness is the ability to reflect, to see ourselves not only as creatures of flesh and bone, but as part of a larger story, a story that began long before we were born and will continue long after we are gone. In the quiet of the night, as your thoughts drift and settle, you are both audience and author, both question and answer, both wave and sea. The journey continues, ever deeper, ever outward, ever into the luminous dark. And so, our voyage through the quiet miracle of how one single cell dreams itself into a brain gently drifts to a close, like a comet tail fading into the hush of space. May this tender unfolding of life's mystery settle softly in your thoughts, inviting peaceful wonder as you rest. Until we meet again beneath the starlit sky of curiosity, return soon for more dreamtime tales that whisper the secrets of our universe.